Our final talk in this session prior to our panel discussion will be presented by Dr. Roy KHALIFÉ from the Ottawa Hospital and Dr. Jerry TEITEL from St. Michael's Hospital. Their talk will be on hemophilia gene therapies rollout. Are HTCs ready?
Thank you. Delighted to be here today and be co-presenting with Dr. TEITEL. I think a lot of the speakers before us anticipated our talk, so we'll see how we go through that. We were just briefly discussing that we did forget to add a disclosure slide. I think we are all living with some conflicts in our lives, but I'll spare you that. So the objectives today are to describe models of care recommended for the delivery of gene therapy, outline the logistics and feasibility consideration of implementing gene therapy, and then reviewing administration and monitoring process. So we've talked about gene therapy being a potential cure for hemophilia, and we've had evidence of long-term expression of factor VIII and factor IX, but we need longer-term studies to further understand what's happening afterwards. But we've seen that recent approval in the US and Europe for different gene therapies, and would it be possible to see it happen in Canada in the next 5 years? Maybe Dr. Lillicrap will come back in 2025 and say, it's here, I told you so. So we need to, I don't have to tell you that Canada is a pretty wide country from coast to coast, up to the north. We have to figure out how to integrate that in our care and hemophilia treatment centers. And it will be complex. Not every center will be able to provide gene therapy, but how can we make sure that there's an equitable approach to every patient who's living in Canada to be able to access a therapy like gene therapy? Now, the important work tends to happen before, but also after the administration of gene therapy. A lot of it has to do with identifying and selecting patients, and some of the speakers have alluded to that fact, but then also afterwards, a lot of work happened in terms of monitoring and for safety and efficacy. We need to pay attention to your needs, your perceptions, your values about treatment options. We've heard from two individuals living with hemophilia about why one opted and why did not opt, and we have to take these into consideration. There are strict eligibility criteria. Dr. TEITEL will go through those a bit later. But also, the field has changed. There's a lot of new therapies, and it's all evolving. So how do you make a decision about which therapy to use? What is best for one individual may be different for another one. So we have to go through that discussion and be able to make a decision based on what each individual will want to target or achieve. The administration process is probably more streamlined or straightforward. I couldn't find a picture of anybody sitting and getting gene therapy, so I'm glad that Luke showed his picture when he was sitting in a chair, but I do promise you that you get a chair or a stretcher. And the process is an intravenous infusion, and there's probably going to be a lot of people watching you that day. And Dr. TEITEL is showing a picture of how many people were in the room when somebody was getting gene therapy. And so Dr. Lillicrap went through the administration process, how it goes through the liver, so I'm not going to spend time there, so we can focus more on the questions later on. But a lot of work will happen afterwards. So monitoring from a short-term perspective to long-term perspective. Immediately during the infusion, there may be risks of infusion reaction. We need to have these protocols in place to manage them. And then looking at the efficacy side, so what bleeding events happen, what factor activity level people are achieving, when can they come off their hemostatic therapies, and then the durability of that expression later on. So there's a lot of blood work that's going to happen, a lot of visits that are going to happen to your HTC, and we're not going to let you go away as easy as some people might think. On the safety side, so there's been discussion about liver toxicity, being on steroids, how long will you have to be on steroids, and what are the side effects related to that, other potential immunosuppressing medications. There is going to be a challenge to your immune system. So there is an immune response that's going to happen to the delivery of that vector with gene therapy. What does that mean to you as an individual, to your immune system? Now, when you're seeing factor activity levels up to the 200, we always get worried about clots, so forming unwanted blood clots. There has not been any reports, as far as I'm aware, of any thrombosis or unwanted blood clots. And same in terms of neurological events. So there's animal studies showing the vector or RNA being present in brain tissues, but in humans, there's been no reports of neurological events, but these questions have been brought up, as well as genotoxicity, as Dr. Liddy-Crab alluded to earlier today. So when we have to bring this therapy to Canada, there's a lot of building capacity that needs to happen, and this work needs to happen now. So David this morning alluded to emicizumab taking 2 years, 18 months, 24 months before getting into our clinics. On the oncology side, so the chimeric antigen T-cell receptors for people living with leukemia or aggressive lymphomas took a long time to establish itself as a therapy in Canada. So we need to start this process now. A lot of education needs, both on the patient side, but also clinicians, lab and pharmacy side. We need to make sure that the center has the biosafety requirements to be able to store and handle gene therapy, so not every center may be able to do so. And if not, then how do we work? What's the workflow that needs to happen between hemophilia treatment centers? How do we coordinate that care to be able to make sure that each patient has equitable access? We do want to make sure that we're learning from all of these experiences, so CBDR or other registries will have an important role will continue to have an important role in that from that perspective. We've seen with emicizumab, CAR T-cells therapy, there's a supply and demand that may play a role as well. So, if there's a high patient volume, not enough manufacturing, how are we going to deal with this? How are we going to make these decisions of who gets gene therapy right away, who might have to wait? One of the models of care that has been described and recommended, specifically driven by European countries, that hub and spoke model. So the hub is the center that delivers gene therapy, and spoke centers are the referring centers and those who will be taking care of the monitoring afterwards. It's a collaborative model of care approach, but we need to establish these partnerships between hemophilia treatment centers if we are to proceed with such a model. And how do you divide that labor that is required for gene therapy? So as I said, the hub may be pretty simple. You have to supply and administer gene therapy. The spoke center has to identify who wants to get or go through gene therapy and take care of the long monitoring as patients go back to their spoke center. In between, there may be overlap, so the informed consent probably needs to be taken care of at different time points, and probably a shared role between both centers. Short-term monitoring, registry management, so data management. So how does that play out in the real setting? And now I'm going to pass the podium to Dr. Teitel.
All right, thank you. So I'm going to continue from where Roy left off with this question about hub and spoke. And my question is the hub and spoke model a good fit for Canada? And I'll make a few points here. First of all, that the hub and spoke designations might imply that there are variable degrees of expertise at different HTCs, hemophilia treatment centers. Now, as you know, we don't have any subclassification of HTCs in Canada. We assume that every HTC must be able to deliver state-of-the-art care to all their patients. And historically, we didn't see the need for a hub and spoke type of model with other new technologies. With the introduction of all the new generations of factor concentrates, the subcutaneous therapies, we all accepted our responsibility to learn how to use these technologies and to support one another in using them. And we do have excellent links for consultation and collaboration well established around the country for specialized testing and for difficult or rare cases. And examples would be regional or national reference labs. Best example would be the National Genetic Testing Lab that David set up in Queen's University. Now, we will likely, though, require collaborative approaches to gene therapy infusion and detection of anti-AAV antibodies. And these are 2 points I'd like to talk about a little bit more. And Alfonso kind of introduced this in his talk because we actually have a wonderful collaboration with our friends at McMaster, with Alfonso and Davida Mattino, because our center, we do enroll patients in gene therapy clinical trials, but we don't do infusions. Our pharmacy, our research pharmacy, is not set up to do it, so we need to use our colleagues. We drive down the road to Hamilton, infuse the patient comes home, and we look after them. So this is for clinical trials. Now, what's going to happen when we get commercial product? Will pharmacies at all HTC sites be able or willing to infuse commercial gene therapy vectors? Well, I'm looking here. I've shown you some of the excerpts from the product inserts from the 2 available gene therapy products. The first one is the Hemogenics products licensed by the FDA. And it says, prepare the product using sterile technique under aseptic conditions, proper engineering controls such as biological safety hoods, and according to institutional policies. Now, if I show that to my pharmacy, my outpatient pharmacy, they may say, that's fine, we can do that. And the bottom one is from the Roctavian product, licensed by the EMA. During preparation, administration, and disposal, personal protective equipment must be worn. So you have to wear PPE and make sure that you dispose of them properly. Well, again, that doesn't sound too onerous. So clinical trials are different in many ways, including this from commercial product. What about then anti-AAV neutralizing antibodies? David introduced this in his talk. Well, this is again from the factor IX product, the Hemogenics product, the top quote. In AAV vector-based gene therapies, pre-existing neutralizing anti-AAV antibodies may impede transgene expression at desired therapeutic levels. But as I think you already heard, the trial from which this was licensed, that led to the licensure of this product, did include patients with neutralizing AAV antibodies. And one patient who had the highest titer did not have a good response, but the other patients did respond. So this suggests that perhaps we won't need to even measure neutralizing antibodies to treat factor IX patients. On the other hand, the Roctavian product, the bottom quote, says that it is indicated for patients without inhibitors and without detectable antibodies to AAV5. So for that product, yes, it seems that we certainly will need to detect or screen our patients for neutralizing antibody. So what about limitations? Is AAV-mediated hemophilia gene therapy a definitive cure, or is it a bridge technology? And remember, we're talking now about the first generation of hemophilia gene therapy. So on the left-hand slide, this is from, I misspelled Johnny Malangu's name, apologies to him, from a paper, these are actually both published in the same issue of New England Journal of Medicine earlier this year. So the one on the left shows the factor VIII levels with time. David showed some of these data already, and the light blue shows extrapolation. And the extrapolation predicted that the median chromogenic factor VIII level would be 5.7% out to year 5. So a lot of patients, perhaps if we extrapolate that further, this will not be definitive cure for hemophilia A. On the other hand, and again, you've heard this already this afternoon, the factor IX gene therapy, much more robust, and we can anticipate that perhaps for many patients this will indeed be definitive therapy. Further limitations. Well, you've already heard this again. Large populations will be excluded from the first generation, the ones that are now in trial and licensed, the first generation of hemophilia gene therapy. Children and adolescents, for very good reasons, they were not included in the clinical trials. We won't go into the reasons now, but that's a big population. What about patients with factor VIII inhibitors, or rarely factor IX inhibitors? And I won't reiterate the question about anti-AAV antibodies, patients with liver disease, Rick highlighted this as one of the reasons that he decided not to go ahead with gene therapy. And patients who lose the lottery for early access. Now, I don't mean that literally, but figuratively, I think it is, in a sense, a kind of lottery in the sense that we have to assume that availability is going to be restricted. First of all, by limitations on reimbursement, and secondly, vector supply. You know, when you look at recombinant factor VIII or factor IX, if there was enough manufacturing capacity, theoretically, we could supply enough recombinant factor VIII and IX for the whole world. Not so easy to do it for gene therapy. Producing these vectors is technologically extremely demanding, and even if there was the desire to produce enough for everybody who was interested, it would be a very, very big deal to do it. It's much harder than just producing the subcutaneous protein-based therapies or recombinant technologies. So our challenges then as a professional community. We have to communicate realistic information to our patients and the community at large, and that information has to differentiate, as you've heard, between hemophilia A and B, the expectations. We need to develop consensus around the need for, the methodology for, and the sites for neutralizing antibody testing. There are, we didn't get into this today, but there are different types of assays for neutralizing antibodies. Total antibodies, anticapsid antibodies, or functional transduction inhibition assays. We need to develop approaches for the management of non-responders, the small number of people who don't respond, and those who lose transgene expression. As you've heard, we can't retreat them, at least with this generation of gene therapy. And we need to develop consensus around recommendations for lifestyle modification, for example, deferral of childbearing. What about use of alcohol? What about medications that may affect liver function? Short, intermediate, and long-term monitoring. Liver function tests, doing ALTs is pretty easy, but it's pretty crude technology. What about liver imaging? What about liver biopsies? What about factor assays? You know, when we presented these, none of the slides that we've shown you this afternoon have shown you what assay was used to measure factor VIII and IX. And the two types of assays we typically use for monitoring our patients, there is at least a 40% difference between them. And we don't even really know which one is more biologically relevant after gene therapy. It reminds me, years ago, there was an editorial that was titled, "Will the real factor VIII please stand up?" And you know, we're still asking that question. And with gene therapy, the question is even more relevant. And finally, we're going to be one other point was data collection and dissemination. And this is something that we have a responsibility to the community, to the whole international community, to collate our data, to collect it, to contribute to understanding and to the development of the science. And finally, we're doing all this in an environment in which other new therapies are being introduced, new subcutaneous therapies, new protein-based therapies. So, and this I think is my last slide, our role in the development of the next generation then, generations 2, 3, 4 of gene therapy products. Participation in clinical trials so far in Canada has been limited, and we haven't had any formal collaboration. It's been kind of a one-off, you know, whichever clinics have been approached or have decided to participate. So I think we might want to consider a collaborative national approach to recruitment into these next generation clinical trials for gene therapies that will involve pediatric patients, patients with neutralizing antibodies, trials using integrating viral vector gene therapies, lentiviruses, retroviral gene therapies, non-viral vector gene therapies, perhaps ex vivo gene therapies, and certainly gene editing trials using zinc finger nucleases and Cas9 proteases. So with that, actually I was going to turn the conclusion slide over to Roy. So come up, you produced the slide, Roy, so I'll let you highlight what you want to, how you want to conclude.
Oh, good.
Take the mic.
I can take the questions.
Yeah.
Okay. So as we discussed, gene therapy will be a powerful approach in hemophilia treatment. We just need to make it right. We need to figure out how to implement this complex therapy into the Canadian healthcare setting and start planning now. So shameless plug, thank you to CHS and Pfizer Care Until Cure grant. We are beginning to look at these questions. You will be receiving surveys. So it's work done by patient partners, physicians, nurses, physiotherapists across Canada to figure out how we can plan and get the roadmap for gene therapy established in Canada.